Hi students, I am Dr. Badrinath. In this video, we will see the mechanisms of drug transport, also called as a mechanisms of drug absorption, drug per. Okay, so actually we will thought that don't think that transport mechanism of drug transport means transporting of these paracetamol bags from Hyderabad to Calcutta to Delhi, not that transport. Okay, transport means drug particles, drug molecules, how it is transported within the body, within the body. Okay, that means that drug transport from gastrointestinal tract into the blood, as well as from the blood into the cells. Two steps you have to keep in your mind. Transport of a drugs from the gastrointestinal tract into the blood, transport of drugs from the blood into the cells, okay, or tissues, cells, whatever it may be. That's why this is also called as a passage of drugs across the biological membrane. Our body consists of number of biological membranes, we know very well. So drugs, how the drugs will be passage, number of gates will be there. Drugs, to show the its performance, it has to cross the number of layers are there in our body. All the layers it has to cross. And also gastrointestinal absorption of the drug. From the intestine, it has to enter into the blood. Okay? Yeah. Mainly three are there. These three you have to keep in your mind. Very, very important. Whenever they will write, they will ask, this question is very important, and definitely they will ask in the examination. Guarantee question it is. 99.9%, not 100% guarantee. 100%. Mainly three, there is a number of mechanisms are there, but comes under the three headings. First one is paracellular transport. It is nothing but, see here, number one, see the pointer, paracellular transport, permeation through the junctions. See here, one I have given. This is the one, transport one, paracellular transport, one. Through the junction, see here cell is there, here cell is there, between the cells there is a junction is there. Through the junctions it passes. Paracellular, here cell is there, parallelly here also cell is there. This is also parallelly it travels. That's why it is called a parallel cell transport, parallel, paracellular, parallel to the cells. Number two is a transcellular or intracellular transport. Intra means within within the cell, like that one meaning is there. Okay, transcellular means transparent cell is there, like that we can imagine. This is the transcellular. See the second number. Second number, see here. Transcellular. Cell is the transparent. So the drug particle process through the cell. Through the cell. Not through the junction. That point important. Okay. Through the cell. Okay. This is the transcellular. Also called as the intracellular. Okay. Permeation through the cell. Third one is vesicular transport. This is also called as a endocytosis. Also called as a corpuscular. Why this is called a vesicle means here there is a formation of an empty vesicle will be there. Vacuum like within the cell it will be created when the drug comes here. So the drug particle will enter into the vacuum. See here. So this vacuum plays a vesicle. This is called as a vesicle. That's why this is called a vesicular transport. This is also called as a corpuscular. That corpuscle. Okay. That's why it is also called as a corpuscular transport. It transfer this vesicle transfer to the another side, so the drug will be delivered. See here. Okay, empty place is, it is neither a paracellular nor a transcellular. It is just like a in between of the thing, like that. Here there is a two things. Now you come to the classification. Paracellular, in that there is no much things. Here you can see the passive transport, active transport. These you have to read it side by side, so that you can understand well. Passive transport, active transport. This passive transport, carrier mediated transport, facilitated transport, pore transport, ion pair transport. Okay, like that four types are there. Each type, first of all, in the page, in the examination, whenever they will give, first you have to write a first page full of this only. Full of this. Okay. So passive transfer. So any of these headings comes under passive only. Any of this comes under active transport only. See, active means active transport, definitely it must involve the energy. ATP, adenosine triphosphate, you know very well. ATP involvement is must in active transport. Very, very standard important point. Okay. In this slide only, most of the concept is there. Okay. So ATP involved is there. Active transport ATP. You can remember matching. A. And if ATP involvement is a direct involvement is there, then you can say it is a primary active transport. Okay. And ATP indirectly it involves, then you can say it is a secondary active transport. Okay. ATP involvement is must. Here direct involvement. Here indirect involvement. Okay. And primary active transport, ion transport, ABC transport is there. Secondary active transport, Symport, Antiport, two are there. Okay. And vesicular phagocytosis, Pinocytosis. Star mark is important. Star mark. First of all, you have to write this entire concept classification in the exam. Later, you have to explain the star mark only. That's more than sufficient. See here, this is a star mark. Here, star. All these star marks are important. That point you have to keep in your mind. Yeah, this is the syllabus of biopharmaceutics as per your university. This topic I have already completed in 3 videos that you can be watch it. And here in this video, we'll see passive passage of drugs across the biological membrane. Mainly they asked in the passive diffusion, active transport, facilitated diffusion, and pinocytosis. These only they have given mainly, but however, we will try to cover all the things. Yeah, first we will see passive transport, active transport side by side so that you can understand very well. Okay. See, passive transport, no energy requirement is there. It is a automatic process, just like a nature. Okay. One side high wind is there, one side low wind is there, pressure. So from so wind moves from high pressure to low pressure. Okay. If you take a rod, okay, one side there is a high heat, another side is a low heat. So what happens? Automatically it transfer from high heat region to low heat region, isn't it? So that's why this is called a automatic process. See here, drug molecules are high here. Number of drug molecules are more. Here, number of drug molecules are less. So passively from high concentration region to low concentration region, it transport. No energy involvement required. Passively. Here, whereas it needs energy, it takes place opposite to that of this. So from low concentration to high concentration, it takes place and it requires the energy. See here, this is called a energy ATP. ATP involvement is must. AA matching, active transport, ATP compulsory. Okay, this ATP is compulsory. It needs the energy. That's why here I wrote this is a needs the energy. Okay, yeah. Same passive transport here, active transport here. Active transport always needs the energy. See, that's why this fellow is lifting. He is using his energy. He is lifting from uh, end from the earth onto the hill. That's why this is called a uphill transport. Uphill transport. Energy is compulsory from low concentration region to high concentration region. Okay, this is a tough job. It requires the ATP. Okay, that's why this is called a active transport. Whereas here, easy job, just a small, just push is enough. One push is there, automatically energy is need not required. From high concentration region to low concentration region. This is a very easy job. Passive transport. No energy is required. Okay. That's why this is called a downhill transport. This is called a uphill transport. These points you have to keep in your mind. Now I made this here. Passive transport, active transport. Okay, same. More common passive transport. Okay, this is a less common. Opposite words. That's it. This is a more common. 90%. See here, P shape of the letter C 9. 90% of drugs undergo. So 9 and P almost matched. Like that you can remember. 90% of drugs undergo. Here only 10% of drugs undergo. movement of a drug, how it will be high concentration to same points what I have explained, same high concentration to low concentration. Here low concentration to high concentration as per the concentration gradient. This is against the concentration gradient. Okay, this is a downhill transport, this is a uphill transport, this is a easy, this is a tough. Okay, since it is a tough, it requires the kinetic energy. Here, since it is a easy job, kinetic energy, Brownian energy is enough. Here, Brownian energy is not enough. That point you have to keep in your mind. What is Brownian energy? That I will explain you what is the Brownian energy, very, very important one. Okay, so kinetic energy, Brownian energy, each and every drug particle which is rotating in our bodies, which is circulating in the blood or extracellular fluid, whatever it may be, it is having a Brownian energy, Brownian motion. Any particle which is in the motion having some kinetic energy. That kinetic energy is called as a Brownian energy, Brownian motion. Okay, that energy itself is more than sufficient for passive transport. Here I will show you what is the Brownian motion. Only the concept of Brownian motion, this is very, very important. It is there in the colloidal chapter, it is there in the colloidal chapter. Okay, and suspensions settlement, that is also there. See, whenever the particle size is less than 0.5 micron, then you can say it is a colloidal particle, less than 0.5. If it is a greater than 0.5 micron, then you can say it is a non-colloidal particle. Okay, particle size. Here what happened, gravitational influence here no. Since the particle is very small, no gravitational influence will be there. Here gravitational influence is there. That's why the particles will be settled down. Since see, the particles are bigger here, all the particles are bigger. It will be settled down. Okay, particle will be settled down and do not move because the particle is bigger than this. Here, this size of particle not at all influenced by the gravitation. So particle will not be settled down and it is always in the motion. Keep on motion like this. It will never settle down. During that motion only, it has some energy. That energy is called as a kinetic energy by virtue of Brownian motion. This is Brownian motion. This is a sedimentation. How the suspension, the particles will be settled down. Okay, this is called a sedimentation. So Brownian motion, sedimentation, both are exactly opposite. Okay, here the kinetic energy of a particle itself is more than enough. No additional energy is required. Whereas here additional energy is required. That additional energy is nothing but ATP, adenosine triphosphate, or it is also called as a cellular energy. Okay, the cells which are present in the membrane will be break down and it liberates the ATP. That ATP will take the particle. Okay, that here. Here equilibrium is achieved. Here equilibrium, see how the particles is moving from high concentration to low concentration. So that equilibrium, same concentration may get. Here it moves against the equilibrium. Equilibrium is disturbed. Here Fick's first law equation we are going to be discussed in the passive transport. Here active transport need no equation is required. Okay. And example, these examples generally they are asking in the competitive examination. In regular exams also you have to write this. Water soluble molecules like a small monosaccharides. Okay, it takes place. Lipids, this point very, very important. Here also one technique is there. Lipids, carbohydrates and proteins here comes here. Glucose, complex sugars. This comes under carbohydrates. Here proteins, amino acids, large proteins will be takes place active. Keep in your mind, three types is there. Carbohydrates, proteins come here, lipids comes here. Like that you can remember. So sex hormones, mainly cortisol and all, it is hormones all made up of lipids only. Cholesterol, that you know very well. Okay, so that's why automatically link is there. Okay, lipid sex hormones, non-ions, like natural, since it is an automatic and natural process, carbon dioxide pressure, oxygen, gases, like a water equilibrium, since it is a equilibrium is achievement, like that. Cyclosporins, very good example for the thing, cyclosporin. Here you can see ions, here non-ions, exactly opposite here. Enalapril, vitamin B3, B6, B3 is nothing but niacin, B6 is nothing but pyridoxine. 6 if you reverse, you will get a P. Like that you can remember, 6P. Okay. 3 means new. That point you have to keep in mind. Okay. After marriage, Wife and husband, you will get a new thing. Third, that is a baby or boy. That point together. So new baby will come like that. Three, like that you can make a trick here. And vitamin C, absorbic ascorbic acid, vitamin C. So B36C, 5-fluorouracil, 5-bromouracil, and methyl dopa, levodopa. Yeah. Now you come to the Ficks law. Ficks first law, very simple law. This is very simple. Here what happened? See here, the first chamber, here high concentration is there on one side and here low concentration is there. So since the difference is more, rate of transport is also high. That's why here I have mentioned. See, concentration of a drug which is present in the GAT, gastrointestinal tract, and here concentration of the drug which is present in the blood. So it is a two difference, double greater than. That's why here high rate of transport. Whereas already some particles are here. So particle will be takes place, but at medium rate here. Here already equilibrium is achieved. So there is no passive transport will takes place here. No passive transport will be completely stopped when equilibrium is achieved. That point you have to keep in your mind. This law says that higher the concentration difference on the both sides of the membrane, higher the rate of the process passive. The drug molecule diffuses from a region of high concentration to lower concentration until the equilibrium is achieved. See here, until the equilibrium is achieved. That is a Fick's law. He only discovered, Ficks is the name of the scientist. He only discovered, and the rate of diffusion is directly proportional to the concentration gradient. Here higher the rate, here lower the rate, medium rate, here lower the rate, or almost zero, you can say. This point very important. If you remember this diagram and this equation, Fick's law will come. This is a actually equation, Fick's first law. This is the rate. dq by dt is nothing but a rate of change of quantity of a drug with the time. dq by dt. That is equal to DAC. AK47 you have to keep in your mind. So your dad has a AK47. DAK. Okay, having a high molecular weight. Like that you can remember easily. This is a concentration gradient. Just now we saw concentration of drug in the GAT minus concentration of a drug in the blood. H is nothing but a reverse. Thickness of the membrane. Higher the thickness, lower the rate. See, keep in your mind all these factors, including this, which is present in the numerator, directly proportional. Higher the D, higher the A, higher the K, higher the concentration, rate of transport is high. Whereas in the denominator, you have one thing. This is always against. Higher the H, lower the rate of transport. Because membrane thick is more, rate of diffusion is small. Like that you can remember. And carrier mediated transport. We come to the second one. Here what happened, there must be some carrier must be involved. That is the main thing. Either it may be a car or it may be a bus or it may be a truck, whatever it may be. Okay, it may be a bus, whatever it may be, anything. There must be involvement of a carrier. There must be involvement of a mediator. Okay, so in active transport, passive transport, there is no mediator. Directly it comes from high concentration to low concentration to high concentration. Here it is not like that. There must be some carrier. Either it may be a speedboat or boat or whatever it may be. See here, or it may be a jet. Whatever, there must be carrier or mediator. Then you can say it is a carrier mediated transport. See, this is the carrier mediated transport. See, this is the carrier, red color. See, drug part, this is a membrane, blue color is a membrane. So, drug, it forms a complex with the carrier. It is called a drug carrier complex. This complex is a reversible, not irreversible. Reversible complex will form. See the shape of the round shape here. This is also, see the groove of the carrier. Exactly it fits. The shape is important. So that what happened, drug carrier complex will, this complex will transfer to another side through the membrane like this. Here the drug will be delivered. So the carrier again it comes back. See here how the carrier is comes back. Again fresh molecule of the drug will be takes place. The same thing here you can read it. Carrier may be enzyme. This carrier, it may be enzyme or it may be any component of the barrier, this barrier. This barrier consists of the carrier. That point you have to keep in your mind. Drug which binds with the carrier, it forms a DCC, drug carrier complex. This is the complex. See, this is the complex. It is a reversible. That's why here it is delivered and again it is coming back. Reversible, non-covalent bond. This is there is no covalent bond is involved. Covalent bond is permanent bond. So here non-covalent bond is involved. It is a structure specific. See the groove, exactly it fits. That's why this is called a structure specific. Okay. And DCC travels another side of the membrane and it dissociate into the again into the deliver. So drug is delivered on the other side, carrier return back to the initial place to pick up the another drug particle so that it comes back. Okay, this is also called as a carrier transport, carrier mediated transport, mediated diffusion, capacity limited. Why this is called capacity limited? I will explain you. Capacity limited, saturation transport, saturation will be get at one point. That is the main thing here. These three terms are almost the same. I will explain you in the next coming slide. Non-linear transport also. This is also called as a non-linear transport. Okay, this is very, very important and a very simple concept. Simple. Yeah, here first three concepts you have to keep in your mind. First one is first concept in this carrier mediated transport. Concept of the false transfer, false nutrients is also possible because any, not only drug particle, any other vitamin or any body particle, if it is having a similar structure, it go and attaches, round shape only, it go and attaches, not only the drug molecule. Okay, any particle, so it go and it delivers to the head. This is called, that's why it is called a false. Instead of drug molecule, another will go and attach there. Okay, so other particles having a similar structure will also transfer. That is also possibility. This is called a false transfer, false nutrition. It is a structure specific only. Okay, if there is any triangle particle or tetraangle particle, no, it will not be takes place. So example is 5-fluorouracil, 5-bromouracil, very important for competitive examination. They will ask you this one. Number two, concept of absorption window. Here see the window is there. See, we know that carrier must be involved here in this transport. So carriers will be limited to at only one spot. See here, this red spot, this is a gastrointestinal tract. In the intestine, only this region is having a carriers. The remaining membrane doesn't have carrier. Nowhere it is carrier. So what happens? See here, this is this part, red part. So this part is only called as a window because from the window only absorption takes place. That's why this is called absorption window. Remaining part is, it becomes inactive. There is no transport from this way. Okay, see here, drug particle will comes here. Okay, it will be taken up. It will be taken up here and it will be transported. Okay, that's why this is called a concept of absorption window. Absorption window is nothing but a place of a GIT, anywhere, small place, from where which consists of a carriers, from that only diffusion takes place. And concept of capacity limited transport. Why this is called as a capacity limited transport? I will explain you. Because here see, initially what happened, rate of transported is very high because it is directly proportional. At one point what happened, all the carriers which are present in that area will be engaged with the drug particles. So drug particles, once remaining what are there in the queue, it has to be just passed into the wastage only. It is a wastage only. That's why at one point what happened, if suppose say there is a number of carriers are 5, only at a time only 5 carriers will be transferred. For 5 drug molecules will be transferred. Only the remaining has to wait or it has to be passed. That's why this is called a capacity limited. Saturation will become. That's why initially rate is more, but once all the carriers will get saturated, this point, this is called a yield point. This is called a saturated point. All the carriers get engaged. No single carrier is empty. In such a case, what happen? Rate will be fall down. It becomes a straight. Any time only 5 drug molecules will be carried. That's it. Okay. Whereas a passive diffusion, it is not like that. Up to the equilibrium, it go on increasing. Passive. That's why this is called as a linear. Passive is called as a linear, whereas here it is called a non-linear. See, linearity up to this point, there is a linear is there. From here, it becomes a straight line. See here, like this straight line. That's why this is called a here linear plus non-linear. That's why it is called a non-linear kinetics. It is described always by the Michaelis-Menten equation. Okay, curve is best described. This curve is best described by the Michaelis-Menten equation or non-linear kinetics. Okay, see, initially the rate of transport is fast and curve is linear initially. Number of carriers are limited. All carriers get engaged at yield point or saturated point. From there, rate of transport becomes a slow and curve is a non-linear. That point you have to keep in mind. See here, that is a third concept of what I can say. Even though you are taking a 2 tablets, it is equal to 1 tablet only. Why means, see here, one particle is engaged here, it is transporting. So the remaining particles, whatever is there, it will be waste only. It will be passed into fecal matter. So even though you are taking, that's why B complex tablets daily one only enough. Don't take 2 tablets and don't take 3 tablets. Even though you are taking 1 tablet, only 1 tablet will absorb. Even though you are taking 2 tablets, only 1 tablet will be absorbed. If you are taking a 3 tablets of vitamin B, only one B complex tablets will be absorbed. It is 2 tablet, 3 tablet, 4 tablet. Absorption will be one. Remaining will be enter into the fecal matter. Okay, because B complex B1, B2, B12. All these things are absorbed by the carrier mediated transport only. There must be a carrier. It is a structure specific. Okay. See, even though drug dose is increased, absorption will not be increased because of the limited carriers. Okay. Hence, if you take a 1 tablet, 1 tablet will be absorbed. 2 tablets, 1 tablet will be absorbed. That's it. Okay. Next is facilitated transport. Facility must be there here. You have to provide one facility. See here, very simple example and very, very important example. Number of times it was appeared in the competitive examination. Vitamin B12, here carrier must be there. Apart from carrier, you should have one more facility. That facility is this IF. That's why this is called a facilitated. Facilitated. See, intrinsic factor. So see, this directly not fits into the shape. That's why intrinsic factor is having a shape of the triangle shape this side, round shape here this side. This is shape you have to write in the examination compulsory. So here on the one hand it catches the B12, on the other hand it catches the carrier. Like that it forms a complex. This complex will be transported to another side. It will be dissociated this side. Like that. This B12 intrinsic factor is very, very important. There must be some facility. That is very, very important. The same diagram only here. See here, passive diffusion, simply it is coming. This is active, uphill. This is passive is a downhill. Facilitated transport is also downhill, same only. But there is some facility of wheels. See here, wheels are there. Here there is no wheels. So facilitated passive diffusion is nothing but a facilitated transport. Wheels must be there, some facility must be there, like that. Some facility is available apart from the carriers. Okay, it operates concentration gradient, high to low concentration, just like a passive. It is a downhill transport. It is a much faster than the passive. See here, it comes faster when compared to this. Okay, much faster. No energy expenditure is required because active only requires the energy. It requires a petrol, active. It doesn't require any petrol. No, here it requires the ATP energy. Petrol, diesel is required here. So limited importance in the absorption of drug. This mechanism is having a very limited. Only vitamin B12 will, and here entry of a glucose into the RBC cell. This is also one of the example. That is in the blood only it happens. Glucose will be entered into the RBC with the help of facilitated diffusion only. Number of times it was appeared. It was my favorite bit. Favorite bit. Okay, intrinsic factor produced by the gastric parietal cells. These are the gastric parietal cells. Okay, and pore transport. Very simple. Pore transport. Very simple. This pore transport, pore name itself indicates, in the name itself there is a pore, hole is there. Pore means hole, like a polo. See here, polo hole is there, like that. Pore means polo, hole is there. So here, see here, small hole is there here. Here somewhat big, here somewhat big. So type of pore, ultra small pore. Only water molecules will be permeated. See blue color, small particle. Here pore size is 3 to this you have to write in the exam. Compulsory, all the points you have to write in the examination. There is no discount. Small pore. See here, medium also will come here. Small pore. Urea, creatinine, glucose, water. Whereas here greater than 200, large pores. See here, big particles also came this side. See the pointer, big particles. Okay. Here you can make this synonym. This is also called as a bulk flow. Very, very important. But this is number of times it was appeared in the GPAT competitive examination. Any competitive drug inspector, analyst, government analysts, number of times. Filtration, just like a filter, just like how the sieve will be there, from the sieve, the liquid will be passed. No, that's why this is called as an bulk filtration, bulk flow, or filtration, convective transport. These three names you have to keep in your mind in the pore transport. Water flux promotes this transport. When this is called a solvent drag. That's why this is also called as a solvent drag. Okay, it is a narrow, small pores will be there. The pores will be filled with the aqueous filled channels. Okay, we know that membranes are lipoidal in nature. In the membrane, lipoidal membrane, small holes are filled with the water. That point you have to keep in mind. So that water, mainly water molecules will be, which are water soluble molecules will be passed very easily. Okay, so key transport for the following categories of drugs, like molecular weight less than 100, mainly because small holes, no, chain or linear type of molecules will also go here, just like a chain, long chain, not bulky, not branched, linear. Okay, linear type, only linear, just like a snake structure, not the bulky. Branches should not be there. That's why it easily passes like this. Small hole is also enough. So less than 400 Dalton, less than 100 Dalton of molecular weight, low molecular size, less than pore size only it passes. Okay, urea, water, sugar are the common examples. Here also you can see one example. Driving force is directly proportional to hydrostatic pressure. Hydrostatic pressure, water, how it moves, along with that, this is also moves. Okay. Renal excretion, very, very important. Excretion in the renal through glomerular filtration rate. Drug will be removed. From removal of a drug from the cerebrospinal fluid, CSF, and entry of a drug into the liver. These three is very, 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 very important. Ion pair transport, name itself indicates ions formation. See, cation forms with the anion. Anion forms a complex with the cation. It forms a neutral complex. Neutral complex will go fast when compared to ions. Okay, very passive diffusion. Here it will be breakdown. Example of drugs like this. Quaternary, this is a quaternary ammonium. Quater means 4, that you know very well. This is a quaternary ammonium compounds. 4 amino group is a ammonium, NH4 low. So 4, this is having a plus mark. So it links with the negative charge. Okay, and it forms a complex. One more example, see here. Here I have mentioned, quaternary ammonium compounds. Sulfur, this is a sulfonic acid. This is also one more example. Propranolol is also one of the example for this. This point you had, propranolol is a basic. It mainly forms a complex with oleic acid. This is also very, very important, and it forms a neutral complex. Neutral complex will be transferred again. This side it will be dissociated. This is ion pair, not ionic. Ion pair must be there. Pair. That is a very, very important word. Okay. So neutral complex, it goes fast than this is ionic diffusion, not pair. Ions directly diffuse. That's why this is called as a electrochemical diffusion. Electrochemical, electric charge will be there. See here, membrane, this is a membrane. Keep in your mind, membrane is always having a positive charge. Membrane is always having a positive charge. So what happens? Neutral atoms will go at higher rate. See the bigger arrow mark. Whereas an anion, it will be attracted by the positive charge and it will be go pass through a medium rate. Whereas a cation, almost there is a reflection will be there. Reflection. So that it absorbed through the slow rate. Because repulsion will be there. Here attraction will be there. Here neutral atoms are always. So neutral atom first, followed by anion, followed by cation. So this you can remember. NAC. NAC is accreditation for giving to the various colleges. This point you can be keep in your mind. NAC. First one is neutral atom, anion, C, cation. That's it. So the positive charge of the membrane, this is a positive charge of the membrane having an influence on the permeation of drug. Neutral molecules permeates at a higher rate. Anions permeates at medium rate. Cations are the slow rate because of the repulsion. That's it. This is a ABC transport or ATP cassette transport. This is a cassette. See this part is called as a cassette. ATP binding cassette transporter. Substrate drug molecule binds with the substrate binding protein. This substrate binding protein plus drug molecule binds to the receptor like this. Then ATP will be open here. The gate will be open, drug will be delivered to this side. Okay, mainly for transporting of a small foreign molecules. Okay, foreign molecules means not from London or not from the British or not from the America. Outside the body is called as a foreign particle. Okay, this is called a absorption. Very, very important. This point, absorption. Example is P glycoprotein is a ABC transporter. This is a one of the example for this substrate binding is a P glycoprotein. Last one is a Symport. Okay, we have a Phagocytosis also, very interesting one. Symport, Antiport, Uniport. See here, by seeing the image itself, you can came to know. Here, what happens? Symport, same side. Symport means same. Both are same. Yes, yes. Keep in your here. Anti name itself indicates one particle will go this side, another particle will come from this side. Uniport, only one direction is there. Uniport. Symport is nothing but also called as a co-transport. Anti-transport is called as a counter-transport, just like a encounter, counter-attack. The energy which is required for transporting of this, it will be get by transporting of this ATP. Here also same thing. If this particle will transfer from here to here, ATP will become. Here ATP will go if it transfers. So both will be matched. Here also same thing. One ATP will be gained, one ATP will be loosened. So both is matched like that. Yeah, vesicular transport. This is very, very number of times it was appeared in the examination because there is a formation of a vacuum. The first diagram I have shown now. This is called as a vacuum, a vesicle, corpuscular. Drug particle will enter into here. So this vesicle will be transported to another side. See here, vesicle, corpuscle in the cell. The drug particle will enter into the formed vesicle. Then only one of the following thing happen. Either it will be transported to another side or vesicle itself, it eats this engulfed particle. That's why this is called engulfication. Okay. And here, there is a three points are there. Phagocytosis, Pinocytosis. Phagocytosis is nothing but a cell eating like this. Phagocytosis. Pinocytosis, like a PO. PO. PO means just like a cell drinking, liquid particle. How he is taking this liquid. Pinocytosis. This is a phagocytosis. Why means vesicle is the same here. If this particle is a solid, this particle, see the pointer here, if this particle is a solid, then you can say it is a cell eating because we will eat only solid particles, we will not eat the liquid particles. Okay, so engulfment of a solid particle, for example say bacteria, suppose say this is a bacteria, this bacteria will be engulfed by the immune system. Okay, pinocytosis, cell drinking, if this particle is a liquid, this particle is a liquid, then you can say it is a cell drinking, engulfment of a liquid particle. Only the difference is here, it is a solid or it is a liquid. Liquid, pinocytosis. Okay, it is a solid, cell eating. Okay, phagocytosis. This is a pinocytosis. Example is polio vaccine. Here I have shown, no, this is a liquid, See here, liquid only. He is taking liquid. It is in the body, it will be in the liquid particles only. The liquid particles will be absorbed by this. Yeah, okay. So everything we have completed in this chapter. No single point is left. This matter, whatever I am providing, is more than sufficient for all competitive examination, for everything. Okay.